Welcome today, everybody, to our Lipid Maps webinar. Today, I'm delighted to introduce Professor Kai Simmons. Kai received his MD degree from the University of Helsinki with board certification in 1964. He then conducted postdoctoral research with A.G. Bern at Rockefeller University in New York. In 1967, he received a PI position from the Finnish Medical Research Council at the University of Helsinki. In 1975, he became a group leader at EMBL in Heidelberg, Germany, and he started the cell biology program, which became the focal point for molecular cell biology in Europe. In 2001, Kai moved to Dresden to build up the new Max Planck Institute for Molecular Cell Biology and Genetics, and this institute is an internationally recognized center of excellence in its area of research. So I'm delighted to introduce him today. I've been a huge fan of his work for many years, particularly recently with the whole COVID epidemic. I think Kai Simmons and work done by him on the viral lipid envelope is becoming really, really important at the moment. And I'm hoping he's going to speak a little bit about this, because certainly something I'm really interested in personally. So his early research was focused on Semliki forest virus that matures budding out of the host cell plasma membrane to become enveloped by the world's simplest membrane. This simplicity made it possible to find out how this membrane was assembled in the cell and how it was built. And this is so important today because obviously the coronavirus that is making all of our lives very difficult right now is also another enveloped virus that buds out of cell membranes. So Kai's work on Semliki forest virus was seminal at the time, and because this virus became a tool to study membrane trafficking, and using this virus, he and his colleagues unraveled how viruses can enter and exit cells. And this is very particular to enveloped viruses, which behave in this very particular way, making them quite unique in this situation. So I think understanding how viral lipid envelopes are formed and can be damaged and can be targeted is really important right now. So from there, Kai Simmons moved on to study epithelial cell polarity using enveloped viruses as tools. These studies revealed the biogenesis of the apical membrane and formed the basis for what we know about epithelial polarity today. His recent research focused on cell membrane organization and function. He pioneered the concept of lipid rafts as a membrane organizing principle based on the phase separating capabilities of sphingolipids and cholesterol in cell membranes today. Important work. Recently, he founded a startup, LIPIDOMICS, that specializes in lipidomics and is developing mass spectrometric platform which can measure health and disease. So I'm delighted to introduce Professor Kai Simmons today. The title of his talk is Why Lipidomics? From Lipid Rafts to Lipidomics. And I'm going to hand it over now.
Welcome to my webinar on why lipidomics, from lipid rafts to lipidomics. Lipidomics, yes. Let's start with some basics. So you all know that the basic unit of life is the cell, and all eukaryotic cells are organized the same way. They have a nucleus, they have the endoplasmic reticulum, they have the post office, the Golgi complex, they have the power plants, mitochondria, they have the lysosomes, and all these cell compartments are surrounded by membranes. And that's where, and this cell membrane is a two-dimensional liquid of lipids and proteins. And the basic unit of this matrix is the lipid bilayer. Now, lipids and proteins, so the proteins, these transmembrane proteins that you see here, 30% of all proteins in the proteome are transmembrane proteins. So a lot of the activities occur in our cells, and many proteins also visit the membrane from either side. So it's a real hub of activity, and that's what makes membranes so interesting. Look at these three lipids here: sphingolipids, sphingomyelin, head group phosphocholine, cholesterol, glycerophospholipids. This is a phosphatidylcholine dioleyl with two unsaturated fatty acids. Now you take these, dump them into water, and then you see how this lipid bilayer self-assembles. It's a simulation by Elpo Vattulainen, and you see here how these three lipids now are organizing by themselves. They form the lipid bilayer. And you can see here now how the bilayer is forming, and when we have it all there, I'll show you which lipids here. So here's the bilayer. It says it's symmetric. That's not the case in our cell membranes. This is cholesterol. And these are the wiggly oleoyl chains from the phosphorylcholine. And these brown ones are the ceramide hydrophobic chains of the sphingomyelin. So the amazing thing is when I started in membrane research, and that's a long time ago, as you heard, the lipids were center stage, but then slowly they disappeared. The DNA revolution had so many things, interesting things going on, DNA, RNA, proteins, and the proteins in the membranes took over. And the lipid bilayer became a boring, fatty liquid. And you can see this, here is the famous Bartlett phosphorus assay. It's needed for quantitating lipids. It's a well-cited publication over, since it was published in 1958. And here you see the fall, first the rise and fall of lipid research. 1959, oh, it goes up to 1982, and then begins the fall. So there you are. But the lipids are coming back. We have a lot of lipids around us these days. There's an obesity epidemic. Look at United States. 1998, mostly blue. The BMI under 20. And Germany the same. And now only 7 years later, United States has an adult population with 40% of the population with a BMI over 30. Look at it here. Now it's red, yellow, up over 30. And this was 2005. Now it's this high. Germany is following, Japan 3.6. But the epidemic is spreading all over the world. And the problem is that obesity can give rise to diabetes, type 2, myocardial infarction, fatty liver, cancer. Obesity causes more cancer today than smoking. And the total cost, if you take from the United States annually, $200 billion in extra health costs, 150 billion of lost productivity. It's a menace. How did we get into this mess? Well, it started early. The fight was between the fat and the sugar camps already in the 1950s. The leader of the sugar camp was John Yudkin, and the leader of the fat camp was Angel Keys. John Yudkin was a gentleman, British scientist who worked on sugars, and here is his book from 1972, Pure, White, and Deadly. But he was overrun by this aggressive Angel Keys, who came up with this cholesterol and fat equals heart disease. And he had a lot of papers which showed that if you eat animal fats, it's bad for you. So cholesterol was bad for you at that time, and fats were bad for you. And we know now that the sugar industry shifted the blame to fat already in the 1950s. They started the Sugar Research Foundation. The whole idea was to make the fats the culprits. And then there was a Senate committee that was headed by George McGovern, and gave then in 1977 recommendations for the American people, what should they eat. And the recommendation was fats down. And what did that lead to? It led to fat-free anything, doesn't taste very well. So corn syrup was added, and that contains a lot of sugar, fructose. And if you look what happened, here is now 1980, and you see how the obesity epidemic is going up. This is the prevalence going up. So fats, downs, and sugars up was bad for you. But now comes the big bang. So Angel Keys died 2005, and his right hand died later. His son went into the basement of his father's house and saw a big chest of data. And those data he took to his father's lab, and this is the paper that came out in a few years ago. And it reevaluated a big experiment for the traditional diet hearts hypothesis. It was an experiment of 10,000 people between 1968 and 73. They were divided into two groups. One group got animal fats and the other group got vegetable oils. And the outcome was, they were followed for 5 years, and the outcome was that those who got linoleic acid, the vegetable oils, they died a bit earlier than those who had animal fats. And also, it didn't fit at all. So Angel Keys put this study into the drawer, and he advised George McGovern, 1975, 76, they started this committee to go on and take away the fats. So that's how it was. Angel Keys was wrong and George Judkins was right. But we still do not understand what mechanisms are responsible for obesity, and we are doing very little to stem the epidemic. Come back at that at the end. But lipid research is coming back, and it's clear that the complexity of lipids must have a reason. So here you look at the glycerophospholipids. You have different head groups: choline, ethanolamine, serine, glycerol, inositol, phosphatic acid. And then you have a different change in the glycerol in SN1 and SN2 positions with different lengths, more or less, one or more double bonds or saturated. And then you can, from the elements we have in our lipids in our body, we can make over 9,000 individual lipid subspecies. And the sphingolipids are also complex. They have a ceramide backbone, a phosphocholine, sphingomyelin head group, or hundreds of different carbohydrate units, glycan units, to make glycosphingolipids. So there's a lot of variety here. And if you look at that's the fatty acid biosynthesis, you see how these complex pathways make the fatty acids that we need. It is clear that these lipids are needed. And now we have the methods to analyze lipid diversity. In the 1980s, when I started, you had three thin layer chromatographic or liquid chromatographic methods. And there, for instance, phosphatidylcholine just forms one band. And we know it can contain up to 180 subspecies. Then came the triple quadruple mass spectrometers in the 1990s, followed by hybrid tandem mass spectrometers. And they now can resolve the whole variety of lipids in our cells. And we know now that cellular lipidomes, cell types, have different lipids, contain hundreds of distinct lipid molecular species. So very clearly, we have to understand what its complexity is good for. And if you look at lipid function in membranes, we know that they regulate fluidity, permeability, lipids and proteins interact, and lipids regulate cell metabolism. They are cropping up all over the place now. And then we have this important concept of membrane subcompartmentalization. Come back to that. And of course, we have the storage lipids. Triglycerides and cholesterol esters are stored in lipid droplets in all cells, but especially in adipocytes. And they play an important role in energy metabolism. And of course, in obesity, the adipocytes just swell up. The lipid droplets become one big droplet that fills the whole cell and makes us obese. So, membrane subcompartmentalization. Rafts, that was the concept. And here is the pictures I saw as a kid, films of logger movies. And you can see here is the raft. It's of course not a liquid, it's solid. And then you have these four guys here, the four loggers, and these are the proteins, and this is now the raft platform. Okay, it's a simile, but that's how it is. And here you see then sphingolipid cholesterol protein rafts. So what's the raft concept? It is, first of all, a principle to sub-compartmentalize membranes in a dynamic way to form platforms where activities can take place. And these platforms are formed by the sphingolipids, sphingomyelin, and the glycosphingolipids, and also by phosphatidylcholine if they are have saturated fatty acids. The sphingolipid the ceramide has stiff saturated chains. And as you know, the lipid bilayer is asymmetric. Phosphatidylcholine and the sphingolipids are in the outer leaflet, and then the other leaflet, in the cytoplasmic leaflet, you have the other lipids, which in this case here form an assembly with the outer and the inner leaflet. And this assembly is held together by weak interactions. The assembly is somewhat thicker than the rest of the membrane, and it's more tightly packed, it's condensed with lower conformational order. But it's a liquid, it's ordered, but more condensed, more viscous. Okay, and you have also proteins in the rafts, GPI anchor proteins on the outside, which has a GPI anchor with two saturated fatty acids which intercalate into the raft. And from the inner side, the cytoplasmic side, you have acylated proteins, many tyrosine kinases, with two saturated fatty acids, which also then intercalate in the cytoplasmic leaflet. And of course, you also have transmembrane raft proteins. We come back to that. So the rafts are dynamic. They associate and dissociate on a millisecond time scale, and they fluctuate, so say they fluctuate. But you can make them, so here you have one phase. But now, if you oligomerize raft lipids or raft proteins, then they stabilize by collective activation and form an extended order membrane nanodomain. And these lipid rafts have many functions. They function in polarized trafficking in epithelial cells and in neurons. They are involved in viral budding. Influenza virus buds out of a raft from the plasma membrane. HIV does the same. Coronavirus does not. I won't really be talking about coronavirus here or COVID. And then you have all the signaling platforms, for instance, in IgE signaling, or IgE in T cell signaling and B cell signaling, where your rafts are activating signaling platforms. And the most important thing now is that the physicochemical basis for the lipid raft concept is liquid-liquid demixing, phase separation. We have two phases, the liquid disordered phase and the liquid ordered raft phase. So why do vinaigrettes separate? Vinegar and oil separate if you let them stand around, and this is phase separation. Two different phases. And you can do the same with cells. You can blow them up into balloons. Here you have A431 cells, and here you see them after 3 to 12 hours in this buffer, and now they are balloons. The plasma membrane is the balloon, and these are the cytoplasmic remains here. And this cell line is enriched in the raft ganglioside GM1. And if you now express a raft protein, it's red, and you blow them up into balloons, this happens at 37 degrees, then you add cholerotoxin, which clusters GM1, it's pentavalent, you get a phase separation. Now this raft protein is in this raft phase. And if you look at another protein, the transferrin, it's a non-raft protein. It now separates from the raft phase. So the transferrin is red, and there is cholerotoxin now green, binding to ganglioside GM1. This is the raft phase, and this is the non-raft phase, the liquid disorder phase. So here you have it. So this is all governed by weak interaction by sphingolipids and cholesterol or saturated glycerophospholipids, and repulsion towards polyunsaturated fatty acids. So this balance of forces can be used to induce phase separation. Now comes the question, if you have this assembly here, which is more condensed, more tightly packed, how can a transmembrane protein be included into this assembly. What makes a transmembrane protein raftophilic? And we were fighting this problem with Ilya Leventhal when he was a postdoc in my lab. And it wasn't sorted out when he was there. But now he's a professor at Virginia, Charlottesville, and he came out with this great paper which shows what is the basis for raftophilicity for single transmembrane proteins, single spanning. So he already showed in my lab that the transmembrane domain length is important. So you remember perhaps that the liquid assembly is a bit broader. It has the length, the assembly of rafts is a bit higher than the rest. So the transmembrane domain length has to be longer than the normal length. And you can see that the transmembrane domain length is corresponding to raft affinity going down from here to here. So one issue is transmembrane domain length. And then he came up with the most important part, and that is phase-dependent lipid protein surface tension drives partitioning. So there is a interfacial energy on the surface of the transmembrane domain. And it's different between when in the disordered phase or in the ordered phase. And this delta G drives the partitioning. And if you see here, the whole idea was that the surface area has to be minimized of the transmembrane domain. So here you see how the surface area is decreasing, and that increases raft affinity. So these were the two principles: transmembrane domain length and a slim surface area of the transmembrane domain. In addition, palmitoylation can increase raft affinity. That's a regulated change. You can take away the palmitoylation, deacylate, and in this way. But now we understand what makes a protein, transmembrane protein, raftophilic. Now you can also worry, how could you get these big raft domains in these balloons? Well, that was due to the fact that the actin filaments, they dissociated. And then when you added chlorotoxin and you got clustering of the GM1, you got all the raft platforms fusing and coalescing. But in the living cell, you have these cortical actin filaments underneath the plasma membrane. So now when you add chlorotoxin, you only get small platforms because they get stuck on these cortical actin filaments. So it's an abortive phase separation, an abortive two-dimensional phase separation. That's what drives raft platforms forming, and they can then easily be dissociated. And this dynamics is very important. You create subcompartments which then can be removed, and there can be thousands of them in one plasma membrane, and everyone is isolated from each other so that activity can take place undisturbed. Now, amazingly, phase separation entering big time into biology. 3D phase separation. This is a concept by Tony Hyman from our institute in Dresden, the Max Planck Institute. He showed with his group that the germline P granules, these are RNA protein granules, are liquid, not lipid, liquid droplets that localize by controlled dissolution and condensation. So you have the dilute phase, a single phase, and then you have a phase separation into a condensed phase. And this is now a general principle. There are articles almost in every issue of the main prime journals where you have clusters of different sorts which are formed by these condensed liquids. You can even form them under the membrane. Here is a raft forming platform, and underneath is a liquid droplet forming. So clearly, liquid-liquid phase transitions driven by weak interactions are general in biology. Simple physicochemical principles govern cell organization and regulation. And therefore, cellular system must be poised close to a phase transition. because otherwise you have to be close to the boundary. Otherwise you cannot move from one to two systems and from two systems to one system. If you are close, it goes without much energy expenditure. So there's a tight control of concentration. And this is important. It means that the lipids in our membranes have to be tightly regulated. And therefore, we put up with Andrei Chechenko a method at the Max Planck, a shotgun lipidomics platform, which is very well suited for a lipid analysis. And we then founded a company, Lipotype, and used and introduced this platform into Lipotype and spent 2.5 years to optimize the whole setup, to automate it, sample preparation, extraction, the whole process, writing new programs to get it all under control, and getting a system which overcomes the challenges in lipidomics. First of all, the reproducibility, the comparability of data. This is one of the biggest problems in the omics field in general, and it's not solved, not for lipidomics either. There's a lack of standardization. No complete standardized solutions are available. Lipidomics has one very, very important advantage, and that is that there are standards available from Avanti. This is not true for the other areas. And it's an interdisciplinary research field. It requires extensive knowledge in mass spectrometry, analytical chemistry, biology and medicine, statistics. It is not possible to just take a protein mass spectrometric facility and make it into a lipidomics facility. It doesn't work. And our performance now is a fully integrated, transferable, high throughput shotgun lipidomics platform. And it has high technical precision, high throughput, 96 samples per machine in 9 hours, fast lipid identification, 10 minutes for 96 samples. And it's not absolute, but it has a very, I'll show you, a lipid species quantification. And it's optimal for large-scale clinical lipidomic screening. And in our target list, we have over 2,000 lipid species. So, lipotype, lipid phenotype, multiparametric, robust readout, blood lipid composition as a signature for health or disease. But let's first look at this. Human plasma samples, quantification of 19 lipid classes, using internal standards in every sample, sensitivity, 1 microliter plasma is sufficient, and we get 300 individual lipid molecules very well quantified. Absolute is in citation marks, but here you see that when we study this, you see over 4 orders of magnitudes for the lipid classes, you see almost a linear response. So when you look at PC, there are of course 50, 60 lipids here, and most of them have to be following this linear response here, but of course not all. So the absolute is in citation mark, but it's good enough for what we need today. Later on, we might need more. And reproducibility, as you can see here, if you have quartiles, the range of amounts in the first quartile is the highest. Almost all the lipids are there. And there you see that the coefficient of variation is under 10%, very good. And only in the fourth quartile, when you're close to background, are you close to 20. So this is really optimal. And intersite reproducibility has been demonstrated. Here is Lipotype versus Nestle Institute of Health Science, which uses our platform, and you can see it's a very nice correspondence between the two labs, site 1 and site 2. Okay, let's have a look now at human plasma. So first of all, let's see what is the variation of each lipid species in samples from Finland to Portugal. Here you see the lipid variation, all data sets. And this is the coefficient of variation now. Here's the lowest, cholesterol is there. And then it goes on and on. And there you see here are the highest. So the highest are the lyso-PE, plasmalogens. There you see phosphorylethanolamine, DAGs, triglycerides, DAGs. We now know which ones vary most. We don't know exactly how this samples all were taken, fasting or non-fasting, but we can see most of them are not changing so very much. So we have this is a very good thing, because it means that lipids are very tightly controlled in the plasma, not only not only cholesterol. And this is, of course, an important thing if you want to use them for screening for disease and health. So let's look at obesity. Now, you don't need lipidomics to see if a person is obese. But let's see, if we look at obesity, can you see the obesity in the lipidome? Okay, here's the first try. We look at we have a PCA here, the principal component analysis. Here are all the samples, all the people with their lipidomes. And if you then have the second and the first dimension, you can see here that the highest BMIs are separating away from the lowest, which are here. So there is a differentiation. So then we had a bigger study. This is from Finris, from Finland, 1,000 population study where we have 1,005 individuals. And now we look at them, we do all the lipidomes, we train the lipidomes, we test them and look for the best model. And then we validate that in another sample from Malmö, where we then see if we can get the same resolution as the model we had in the bigger study. And now we look for the body fat. Here's the BMI, here is the waist-hip ratio, and here is the body fat. And this is the normalized mean absolute error. And you can see, and the smaller it is, the better. And the body fat is predicted best. And I just summarize. Body fat percentage, waist-hip ratio are better predicted than BMI by the lipidome. And modeling of the BFP is good with 8% error. We can say we can predict the BMI with 8% error, the BFP, the body fat percentage, with an 8% error. And it's the lipid species detail that is needed for accurate modeling. We have to have the subspecies and the species in the with the resolution of the mass spectrometric analysis in order to see this correspondence. And what important here is outliers have with clinical profiles that predispose for obesity-related diseases. So here you see here is the observed BMI, predicted BMI, and here is the line. And those that are on the line show that we can then, from the observed BMI, observed lipidome, we can predict the BMI. But look at these here, and look at this area here between 22 to 30 BMI, just under 30. And these here are the predicted lipidome gives a BMI which is higher than the observed. And this is those that have clinical profiles. So it's bad for you to have a lipidome which predicts a BMI higher than it is. So it means that the metabolic status of the person is somehow reflected in the lipidome, and therefore it could be that we could use we could stratify obesity to predict future health risk. Now, can plasma lipidomics differentiate between health and disease? Yeah, there's now an abundance of studies that demonstrate that this is the case from Lipotype and others. And I won't go much into this, but I will show you one example from Malmö, from Sweden. This city of Malmö, there they had a big Malmö diet and cancer study, and they collected 30,000 samples. They had 30,000 people giving blood and being tested, and they got diet, they got many smoking, drinking habits, and the samples were collected in between 91 and 96, and then they put into -70 and were kept in the freezers, not done much with them. And 2014 we got these samples and analyzed them. So we had screen with 2,800 healthy individuals. We didn't do all 30,000. And the plasma lipidomes were analyzed 2018. Sorry, this means that they were followed. We know exactly what happened to all of these people up to 2014. We know it now to 2018. And let's look at alcohol per day. Okay, so here is the whole the alcohol consumption in grams per day. This is the training set and the test set. And we see here that here is the fit, and we have a break in the curve at root mean square error. It changes. So between those who did not drink and those who were drinking. And so we had the control group. We knew people who didn't drink alcohol at all. And then we see here at an RMC of 0.74, we get a break. And this equals 2 grams per day. Look at this, 500 milliliters of beer with 5% alcohol, that's 25 grams. This is nothing. Test the same. So there was recently, within the last year, a big meta-analytic study of alcohol drinking, and the authors came to the conclusion with 100,000 of people that even a glass of wine every evening is bad for you. Well, this is less than a glass of wine, and I think the only real reason for this is that people do lie about how much they drink. I think 1 glass of wine, red wine every evening is good for you. Let's look at smoking. So smoking. Here we can now take people who never smoked, and we can really match them. And then we look, what can we identify smokers? And yes, we can. Training set, test set, and the cross-validation. So we can identify 77% of those who never smoked, and 74% of those who smoked. So it's a pretty good prediction, and it's a signature that gives us this prediction. So our working hypothesis is that the blood lipidome, plasma lipidome in this case, is a generalized measure of the metabolic status. We'll come back to it. Interestingly, the plasma lipidome has predictive power years before disease incidence. So here is the Malmö study again. So we knew exactly which were healthy at the start. Then we know which people who got a disease, 48%. And these were the ones that had diseases at the starting point. So here are the incident conditions. The biggest disease category was diabetes type 2, almost 600. And then coronary event was the second. So let's look at these two. So we know now, we took the sample, they were healthy, and then these people got coronary events, and these got diabetes. Let's look at diabetes first. So we now we predict the risk of becoming a diabetic type 2. And here you see the controls, and here you see the diabetes ones. And you see here a shoulder for high predictability. 143 were highly predictable. 84% here, highly predictable. And look this here. No correlation of the diabetic prediction probability and time to diagnosis. There was no real. It was there all the time. And then we have a lipidome signature, which is here are the values that get lower in the high risk signature, and these are getting higher. I won't go through them. And here's coronary events. Same thing again. Here is the shoulder, 109 with high predictability, 85% here. And the same thing, no correlation between a prediction probability and time to diagnosis. So it's a little like blood pressure. Blood pressure can be there quite much, many years before you get the disease. And a lipidome signature of high coronary event predictability. So in diabetes type 2 and coronary disease, the plasma lipidome can years before incident disease exhibit a risk signature. Now comes the big question. How could the blood lipids reflect what goes on in the whole body? It's not in the blood that the disease is. So the lipids that we are analyzing are carried by the plasma lipoproteins. So if you take the chylomicrons, the VLDL, and the LDL, they all have the same structure. It's a monolayer of lipids, proteins, apoproteins, and then there's an internal core that contains the triglycerides and the cholesterol esters, the hydrophobic core. Also, lipid droplets in the cytoplasm have the same structure, a monolayer, proteins which stabilize the whole thing and makes it assemble into a lipid droplet. So in the endoplasmic reticulum, these are all made. The chylomicrons are made in the gut. VLDL is made in the liver. And in every cell, the lipid droplets are made in the endoplasmic reticulum. They are then extruded into the cytosol, whereas these are in the lumen and are secreted into the Golgi complex and taken into vesicles which carry them to the surface and they plasma membrane, and they are then exported. So chylomicrons are made in the intestine, VLDL in the liver, and then they move the VLDL with lipases. It gets smaller, and in the end, the low-density lipoprotein is formed, which is taken up by peripheral tissues over LDL receptors. And the chylomicron remnants are also with lipases made smaller, and then free fatty acids are released that are taken up by adipose tissue and other tissues. So LDL uptake is by the lipid LDL receptors. It forms a coated vesicle, and these coated vesicles then transports the LDL into the lysosomes, where the cholesterol and the lipids are digested. Cholesterol is not digested, taken up and goes to the endoplasmic reticulum, and these lipids that came with the LDL are then used to produce even to some extent new lipids. Let's look at HDL. HDL is mostly made in the liver. There are different types of HDLs, but also some in the intestine. And these are secreted in a very lipid-poor form. It's very little lipid. And then they get some lipid through binding to the ABCA1 protein in the plasma membrane, in extrahepatic tissues in the periphery, and they take up cholesterol, and they take up phospholipids, sphingolipids. And then they form a disc, and this forms a sphere. It's shown here much clearer. Here's the lipid-free, and there's this disc. This is a bilayer of lipids, and the bilayer is solubilized by the ApoA, which binds to the perimeter of this disc and makes it hydrophilic. And then the LCAT esterifies the cholesterol, and in the bilayer membrane, bilayer leaflet, and you form a sphere, and this is taken to the liver. So these HDLs and LDLs and chylomicrons and VLDLs have been emphasized as being cholesterol carrier. They carry cholesterol for the VLDL, LDL to the periphery, and the HDL takes away cholesterol from the cells. But in fact, they are not only taking away cholesterol, HDL, they take away lipids from the plasma membrane. And the LDL delivers not only cholesterol, but also lipids into the lysosomes. So how about it? Could it be that plasma lipoproteins function as a surveillance, delivery, and retrieval system? They could function as nanosensors to regulate lipid homeostasis. Think about it. You have these lipoproteins that circulates to every corner of our body. And in this way, they see what's going on everywhere. And they are there as a way to regulate the cellular lipid compositions. Because they have to be tightly controlled, not only by synthesis, and by the internal mechanism, by membrane contacts and what have you. And it could be that this really makes sense, that the blood lipidome might serve as a measure of the generalized metabolic status. So here we are. I'm starting to get to the end. So very important is our slogan here. Lipidomics for a better life. What are we doing for a better life? Well, here is a new RD project we have. We want to establish a new health measure, dry blood spot tests. So we will just a kit for home use. You just take a diabetic lancet, get a piece of blood on a paper which has been treated. And then we have papers where the lipid composition stays the same for almost a week. And otherwise it would oxidize and change. So the blood lipidome, a fingerprint of my health status. Sampling the blood at home, and then the full blood lipidome is measured by mass spectrometry. This is our goal. Could this be used for prevention with lipotype for personalized health? Can we help to stem the obesity epidemic? Well, we hope so, and we work for that. So I summarize now why lipidomics. After years of neglect, lipids are back. But still, the know-how of lipids around the world is not great. That's a great area for young scientists. I think you have something that will be, it's almost virgin land today, because we have so many new methods, we have so many new ways of looking at things, and little work has been done in this direction. They cannot be ignored anymore because they play essential roles in cell membrane function and in cell metabolism. And very importantly, for that, we now have powerful mass spectrometric technology. We use Orbitrap QX actives, which are great for lipidomics analysis. And with those, we can measure lipid composition comprehensively, quantitatively, and reproducibly on an unprecedented large scale. And only by analyzing lipidomes at the molecular lipid species and sponge species level, subspecies level, are they informative. And lipidomics has the potential to deliver multi-parametric signatures for disease, but we are not there yet. And based on the working hypothesis that the blood lipidome reflects the metabolic status of a person, it might be possible to define personal health signatures by lipidomics. I'm at the end. Of course, I have to thank the whole Lipotype team, plus Andrei Shevchenko, who set up the shotgun method at MPI at the Max Planck Institute. And our most important collaborator has been Ulle Melander from the university clinic in Malmö, and Celine Fernandez, and then we have other collaborators around the world. So I stop here and thank you.
Thank you, Kai. That was excellent. We have a whole series of questions in the chat box, but first of all, I'm going to ask you a question, and that is, you know, the seminal work around lipid rafts is.
So you can
yes,
see
do. Okay, you can see better than I. I'm just looking like somebody wearing goggles in the dark here, but so you know, following on. Yeah, following on from the work on lipid rafts, do you think there's other micro domains waiting to be discovered in cells?
Yeah, I think, you know, that that's why the lipid bilayer and that forms the matrix in our cell membrane is so enormously interesting, because it has so many different lipids there, and that's the product of evolution. So they are there for a reason. And it cannot be, I mean, rafts is one important thing. It uses this diversity, but it cannot be the only one, the only system. So I think that there are much more to be learned. The problem with these domain formations, which are glued together by lipid-lipid interactions, is that they are weak, these interactions. And therefore, of course, they are not so easy to get your fingers on. to see them. But I do think that when we now start to look more and more at signatures and see enrichment of different signatures coming up in different states, we can see some physiological meaning in these signatures. And one more thing is that one of the big holes in lipidomics is that we don't have a bridge to proteins, proteomics, and genes. And this, we are trying to fill this with CRISPR methods, and I would hope that more labs around the world do the same. And when we have that, then we are unbeatable.
Yeah. So I think there is an answer. There is another potential microdomain, which is when platelets are activated and the PE and PS gets externalized, it seems to localize on ring structures on one end of the cell in order for coagulation proteins to bind. So that may well be another one where there's some level of control. And I think we've got no idea how that's controlled either. So I'm going to move into the chat. I think we've realized that phone-in callers we can't unmute. So I'm going to call out a question from Hamdi Awad, who's asking about changes in lipids in the ischemic brain and the spinal cord.
Sorry, lipids were in
yeah, changes in ischemic brain and the spinal cord. What sort of changes?
I don't think I can answer that question. I mean, I think in general, I mean, the brain is, of course, a very important thing is that they have lots of glycolipids and gangliosides, which had not been analyzed very well. I mean, that's still waiting to be done. I could not say you exactly what would go wrong. We have never tried.
Okay. So we're going to take a question from Anna Takchev. Are you able to speak? Anna, are you there?
Yes.
Okay, go ahead. Ask your question.
We observe a high percent of variance explained for the first PC component in blood plasma lipids, and we see that in our analysis too. And I was just wondering if you have some sort of understanding of what the underlying factors are, if it is maybe body fat percentage or maybe some other metabolic health status, or whether it is actually unrealistic to try to find a simplistic factor that could be attributed to this variation.
So once more, what PC was? I didn't get what the molecule was.
Right. No. I meant the first component of the main component of variance.
Sorry. Okay. I mean, yeah, you can see these things. I mean, this is one of the big problems now that we are in the beginning of this analysis and the biological, since it's very difficult to. There's so little data on which species and subspecies are made by which enzymes. It's very difficult to get the biology because of course we have much more information if we can make the bridge to proteins. And that's why it's so important that we now, the idea would be to knock down every protein that has to do with lipid metabolism and then do a lipid with CRISPR methodology, and then do an analysis of which lipids are changing. Very important is that lipids, like in the rafts, but maybe in other situations as well, work as collectives. So there are some collectives that they are. There's a lot of interaction between the lipids, which we have not yet really explored. Just the beginnings are known. And therefore, questions like yours are not easy to answer.
Okay, thank you. So we've got a couple of questions from Maverick Ma, who's asking about membrane fluidity and rafts.
Membrane fluidity of rafts.
Yeah, Maverick, can you unmute yourself there? You should be able to. Okay, go ahead.
Hi, Simon.
Go ahead, ask your question.
Hi, this is Maverick from Taiwan, and this is now is almost 2 p.m. 2 a.m. Almost.
That's nice of you to say up for my.
Okay, so my question to you is that because you mentioned about the lipid raft and also the membrane fluidity influenced by the character of the composition of lipid. So what's your comment whether the better membrane fluidity cause to membrane protein function better, or or the opposite?
Well,
And
the whole
the
raft,
same question is that whether the membrane fluidity influence the protein stabilized on the membrane raft.
okay, good question. I mean, clearly the whole concept of a lipid raft domain, a liquid order domain, means that it's more condensed, more viscous. So it has different material properties. And that's a very important thing, even like in these condensed liquids in the cytosol. Also, these condensed water, condensed liquids there have different properties, which we don't really understand very well now. And the same goes for membranes. But clearly, that's the idea that a protein outside the lipid raft, and then you form rafts, and it goes into the raft, has raftophilic properties, then changes its conformation because of the fact that it came into this new liquid.
Mm-hmm.
So that's
so
one.
you are more, okay.
That's
Sorry.
one possibility. There can also be specific interactions with sphingolipids or specific lipids. The whole question of protein-lipid interactions is still in the beginning. I hope one day that one can begin to see by cryomicroscopy really the lipids which are around proteins in such a way that you can start to look which ones are really bound there, and you could get that from structural biology. There's a hope that this is possible, but not, and some results in that direction has been produced, but we still have a long way to go. So it's not so simple, of course, to measure the binding of proteins to a lipid because you have to do it in a lipid bilayer, in a bilayer state. But also this starting is probably something that can be done and will be done, and then you will know what kind of lipids like to be close there. We had this whole question of this was done before, but that all the methods that were done at that time, they were too crude to really give us information, because we had to use methods which did not.
Okay, great, thank you. So continuing on the same theme, we've got a question from Snigdadev Das, again relating to plasma membrane leaflets. Snigdadev, there you go. Ask your question.
So my question relates to the asymmetric nature of the plasma membrane, more specifically the asymmetric present in between the leaflet, outer leaflet and the inner leaflet.
Yes.
So Ilya Leventhal has recently shown that the chain length and the fatty acid saturation differs very much between the two leaflets. So is it possible that in disease conditions, this thing gets disrupted?
Yeah, I think that if we look at the lipid composition, let's say now the plasma membrane, which will be different in different cell types. I mean, some lipids are the same, but the compositions are different. Then it's the lipid composition in the outer leaflet and the inner leaflet. As you know, the cell spends an enormous amount of energy to keep these leaflets asymmetric, and the lipids that are in the outer leaflet, so to say, they are in the lipid raft, they are the dominating leaflet, and they, so to say, poke into the inner leaflet, usually often into phosphatidylserine, and also a species of phosphatidylethanolamine. So here you have possibilities of now with simulation of lipids, you can start to look at this in more detail because it's so difficult to do it in any other way. So the whole repertoire of methods you have to use to get into this business is that you also have to do simulations of lipids and membranes and proteins.
Okay, thank you for that. So we have a huge amount of questions, but there's no way we're going to be able to take them all. But I'm going to let one last person ask, and that's Oswald Quahenberger from UCSD. Are you there, Ozzy? Can you unmute yourself?
Yes, I'm here. This is Ozzy Quahenberger. I really enjoyed the talk. It was very informative. I have a question with regard to your lipid signature that is predictive for future disease. That is the plasmid lipid signature.
Yes.
The plasmid lipid signature, or the plasma lipidome, is by and large a reflection of the diet. You know, the kilomicrons, those are your dietary lipids. Kilomicrons go to the liver, they get metabolized, and the lipids are repackaged and secreted as VLDL. And so what you really measure in the plasma is the dietary lipid. So would it be possible that rather than it being a predictor for future disease, what you measure is really the diet of that particular person, and that person just has a very bad diet, and that over years will then lead to diabetes or heart disease?
You are, I mean, it's a very good question. I think it's not correct what you say that all the lipids in the plasma reflects the diet. That was exactly, we know when we start to look now at diet, we know which lipids are influenced by the diet. That's not only a small number of them, but we know which ones. So it's both diet and the metabolism in cells and tissues that will decide on what goes wrong. So I think it's yes, you're right. Diet plays an important role, but from this study of thousands of different individuals, we can see which lipids are varying, and we also have looked at if you give a diet to them, we see which lipids are changing. So it's both, and I hope in the future that we can say we can get more information and saying what is the effect of diet and which diet, and we can see signatures of diet, and maybe the microbiome will also be an important issue here. So when you combine lipidomes and microbiomes, you can start to see differentiate in these signatures for different mechanisms that could be behind them.
Okay, thank you.
Okay, yeah, thank you. So that's been a fantastic discussion. I think we're going to need to draw to a close because we're gone 6 o'clock, but we had something like 48 questions there. We'll try and archive them and get them sent over to you, Kai, so you can have a look at them later and see which we can answer in person. But you know, delighted that everybody was able to turn up, and thanks particularly to people from the Far East who were up at 2 a.m. That's really phenomenal, and really enjoyed your talk today, and I'm sure I can speak on behalf of the audience to say that. So thank you very much, Kai Simmons.